Good afternoon. Thank you for joining us this afternoon and welcome to the NEHA webinar on PFAS implications in drinking water. In April of this year, the EPA released its latest regulations for the chemical PFAS, perfluoroalkyl substances. The PFAS family of chemicals, probably the best way to identify it. For drinking water, that new proposed standard is going to be 4 parts per trillion. Now, PFAS chemicals have been around for 70 years, but it's only been recently that it's been found that this family of chemicals have come under greater scrutiny due to its implications on the impact on human health. NEHA is pleased to have 2 experts from NSF International with us today to speak on PFAS and attempts to limit and control its effect on human health. We have Kyle Postmus, Senior Manager, Building Water Systems. Kyle's been with NSF for 11 years in working on water treatment industry and serves as the Global Senior Manager of the Drinking Water Treatment Units Program, overseeing the certification of residential water treatment devices and municipal filters. He's been with NSF since 2013 and is an active observer of their joint committees on water. Bradley Lampe is also with NSF. He's a toxicologist there. He's been with NSF since he graduated from the University of Michigan School of Public Health in 2007. And over the past 10 years has experience in chemical risk assessment and currently specializes in multiple areas in developing criteria for drinking water contaminants, for quantitative structure activity relationship-based assessments for pesticides and pesticide impurities, and risk assessments for extractables and leachables from medical devices. He serves as a secondee to the World Health Organization, providing technical expertise and co-authoring sections on the WHO guidelines for drinking water quality. With that, I want to say thank you on behalf of NEHA for joining us today. And with that, I'm going to turn it over to Bradley. Brad?
Well, thanks so much, Doug, for the kind introductions, and happy to be able to participate in today's webinar. So I'll just start by giving you a little bit of an overview of how this presentation is structured. So I'm going to start by just providing some basic background information on the PFAS compounds, just give you some definitions, some structural characteristics, and then some just a brief overview of some of the toxicological properties. Yeah, just some of their characteristics. And then after that, my colleague Kyle will give you a more in-depth overview of how the EPA's regulations and their recent actions affect our own activities here at NSF as it relates to our standards. So with that, Kyle, if you wouldn't mind advancing the slide. Thank you. So authoritative definitions for the PFAS compounds, or the per and polyfluoroalkyl substances, have been provided by both the OECD and US EPA. I'm aware that there are other definitions out there, but these are just 2 of the authoritative definitions that we sort of go by. So the OECD defines a PFAS compound as a compound with at least 1 fully fluorinated methyl or methylene carbon atom without any of these other elements, hydrogen, chlorine, bromine, or iodine, attached to it. So that's more of a general definition. And then the US EPA, under the Toxic Substances Control Act, or TSCA, has a little bit more of a detailed definition. So they define these substances as a substance that contains at least one of the following 3 structures. So as shown on the slide here, I won't read through those, but you can see that EPA's definition is quite a bit more specific. On the right, we show you a couple of representative structures. So you may have heard of PFOA. That's one of the representative PFAS compounds, P4, or perfluorooctanoic acids. So that is a perfluoroalkyl substance, and you can see on that carbon chain, all of those carbon atoms, except for that last one towards the carboxyl group, is fully fluorinated. Whereas another PFAS compound called octafluoropentanoic acid would be a polyfluoroalkyl substance. So you can see in that one that the one on that tail end, the carbon on that tail end of the chain is in fact not fully fluorinated. So that's just a distinction between the type of PFAS compounds we're talking about. Next slide. Okay. So the term PFAS, again, as you're probably aware, the term PFAS encompasses a wide range of different chemistries, structures, and compounds. If you go to the EPA's website, they have a list of reportable substances under TSCA's definition of PFAS compounds. And if you go to the link to that list under the EPA's CompTox chemicals dashboard, you find that the number of chemicals that fall under the EPA's definition that the EPA has, again, included in their authoritative list is over 13,000 chemicals. Next slide. So the scope of what we colloquially call the universe of PFAS, it encompasses, again, a wide range of compounds, including the perfluoroalkyl acids, or PFAS. So the perfluoroalkyl acids include PFOA and PFAS, which are the two most well-studied PFAS compounds. But again, this is just to really emphasize that those two compounds are only just a tiny subset of the whole universe of PFAS compounds. So some of these are very well studied from a toxicological perspective. Some of these have absolutely no data at all. So that just to give you an idea of the scope. So the PFAS also include the perfluoroalkyl ether acids, which include, if you may have heard of the Gen X compounds, which were intended to replace PFOA. So we have the perfluoroalkyl acids, the polyfluoroalkyl acids as well. And then there's also quite a few PFAA precursor compounds. So some of these PFAA compounds are actual degradants to various precursor compounds. And so again, this is just to give you an idea of the scope of what we mean by PFAS. Go ahead to the next slide. Okay, so the perfluoroalkyl acids, such as PFOA and PFAS, I mentioned that they're often terminal degradation products and that they have several precursor compounds. So for example, on the left, that's just an illustration showing that PFOA is a terminal degradation product of the fluorotelomere alcohols. So a lot of this contamination with PFAS and PFOA isn't necessarily from direct contamination of these compounds, but contamination from these precursors. PFAS is shown on the right, is a terminal degradation of perfluoroalkyl sulfonamidoethanol. So again, PFAS, PFOA can exist in the environment as a degradant, often as a degradant from contamination of one of these precursor compounds. Next slide. Okay, so it's no secret to anyone who works in public health that the PFAS compounds are associated with a wide variety of concerns related to human health. These health effects have been very well studied. I mean, the PFAS compounds, particularly PFAS and PFOA, are some of the most well-studied compounds I've seen as a toxicologist. These health effects have been studied in both human populations and in laboratory animals. You can just see from the slide, they encompass health effects related to reproductive toxicity, decreased fertility, increased high blood pressure in pregnant women, as well as developmental effects or developmental delays, as shown here. The PFAS compounds, again, particularly PFOA, PFAS, have been associated with increased risk of tumors, particularly tumors of the prostate, kidney, and testicular tumors. Also of note, the PFAS compounds, again, this is particularly with PFOA, PFAS have been associated with immunosuppression, and this has been studied in human populations in which it's been suggested or shown that there's a correlation between exposure to these compounds and reduced efficacy of these effectiveness of these vaccines in not necessarily highly, but exposed populations that have been known to be exposed. Also, PFAS compounds are certainly of concern with respect to potential acting as an endocrine disruptor, also associated with increased cholesterol levels and risk of obesity. So it's almost like, what can these compounds not do in terms of toxicity rather than what do they do? I mean, they've been associated with such a wide variety of health effects. Also recognizing that, I'm not sure that any other compounds have been more studied from a toxicological perspective than these compounds. So now, recognizing all of this, the US EPA has spent a lot of time and resources doing risk assessments on these compounds. And in 2022, they came out with their proposed health advisories for several PFAS compounds, again, most notably PFOA, PFAS, but also some other PFAA compounds as shown here. So for example, the health advisory for PFOA is 0.004 parts per trillion or 4 parts per quadrillion. That is a level that the EPA is saying is unlikely to be associated with any health effects as a long-term exposure. PFAS has a corresponding health advisory of 0.02 parts per trillion. So I should mention that these health advisories are intended to be just that. They're advisories, guidance that the EPA puts out, and they do, unlike the EPA's MCLs, these values do not have any specific regulatory force. So the EPA is not mandating that levels come down to these levels per se. Again, these are health advisories, but they should not be confused with the MCLs, which we'll get to in a later slide. Okay, you can advance the slide, please. The PFAS compounds obviously also have environmental health concerns associated with environmental contamination. They're called the forever compounds for a very good reason. That carbon-fluorine bond is one of the strongest bonds in chemistry that we know of associated with organic compounds. These bonds limit and prohibit environmental degradation. So we know that the PFAS compounds are environmentally persistent. Some PFAS compounds are known to bioaccumulate or bioconcentrate. So there's a brief box down there that provides definitions of BAF and BCF. There's bioaccumulation factor and bioconcentration factor. With the PFAS compounds, the bioaccumulation factor tends to be more relevant than the BCF. So you can see that the BAF to BCF ratio tends to be greater than 500. So that just reflects the known characteristics of the PFAS compounds, that they are very well absorbed into tissues from environmental media, particularly soil and sediments. So they're very proficient at being released from these media, the soil and sediment into tissues, more so than migrating from water into tissues. So you can see on the right, the PFOA BAF is 2,000. The BAF for PFAS is greater than 3,000. So again, these again reflect their high ability to bioaccumulate from environmental media. Next slide. Another distinction from a chemistry perspective to make with respect to the PFAAs is this idea of long chain versus short chain. So for the perfluoroalkyl carboxylic acids, the long chain compounds are those with 8 carbons or greater, or 7 or more perfluorinated carbons. So you can see that on the left, the bottom left there with that C8 compound. That's the perfluorooctanoic acid. So that's an example of a long chain compound. And then on the right there, you have a C6 perfluorosulfonic acid. And so those sulfonic acid PFAS with 6 or more perfluorinated carbons can also be considered a long chain PFAS compound. And you can advance, it should show, there we go. The short chain compounds are basically the compounds that are not the long chain. So they don't meet the definition of the long chain. So again, on the third structure from the left, the C7, that's an example of a short chain perfluorocarboxylic acid. That one has 7, I believe. That's a C7 compound. And then on the bottom right, there are the C4 compound, the butane, the perfluorobutane sulfonic acid compound. That one has only 4 carbons in the chain. That would also be an example of a short chain PFAS. Next slide. So why is that important to make that distinction between long chain versus short chain? Well, one example of why that could be considered important is related to this idea called half-life, or half-life of elimination. So in this table, I'm showing you half-life data from these are actual from human participants, studies of human participants that are known to be exposed to PFAS compounds, many of them through their occupation. So the half-life of elimination has been studied in these participants. And so what we mean by half-life here is essentially a serum half-life, or the time it takes for your body to eliminate 50% of that absorbed dose of PFAS. So if you look at some of these long chain compounds like PFOA, PFAS, and the PFHXS, which is perfluorohexane sulfonic acid, you can see that the half-lives of elimination have been shown to range from about 1.8 to 5.3 years in some of these populations that have been studied. Just a very long half-life of elimination. Another reason why we refer to these as forever chemicals. They just persist in the human body. And you can advance the slide. I think there's another, advance it again. And so you can contrast that with a short chain PFAS compound. So some of these, you can see that the half-lives of some of these shorter chain compounds can be measured in days. So that the length of that carbon chain makes a very big difference in terms of how long we predict that chemical to be retained in the body. Next slide. Okay. So this is the part of the presentation where we'll switch gears a little bit, and we'll talk in a little bit more detail about how the EPA's current activities with these compounds and how that affects NSF's activities related to our standards. And so, Kyle, I'll hand it off to you. Thank you.
Excellent. Thank you, Bradley. So just a quick orientation on who NSF is and what we do and kind of what our standards are aimed at achieving. We are a standards writing agency, so we host a joint committee of industry professionals, public health regulators, industry professionals, manufacturers, researchers, and they all get a vote on what ends up in our standard. So we have 2 standards that we'll be discussing today. And the first is Standard 600, which basically adopts all of the EPA values or the toxicological limits on certain chemicals and kind of codifies them into our standards. So historically, we use EPA's regulated limits for those contaminants that have regulated limits. And if there isn't a regulated limit, we do toxicological studies to develop a limit. So now that the EPA has established these values, they are being worked on by the Joint Committee to be incorporated into our standards. There's a ballot being considered and discussed to add these to the standards. And then from that, we can start evaluating products that come into contact with drinking water against this criteria. So on the screen now is an overview of the EPA's rule and the associated levels. So for PFOA and PFOS, it's got an MCL of 4 parts per trillion. And then these other PFAS have been combined into something called a hazard index. Essentially, the hazard index groups them together and performs a calculation. And if the ratio is greater than 1 for that health index or hazard index, it doesn't meet the requirements. So each of these variants are identified against their particular requirement. And when you add that up, if it's greater than 1, it doesn't meet the requirement. And here's a little bit of an example on that, how that hazard index calculation works. So when you're evaluating the drinking water, you would monitor or analyze for each of these different PFAS. If any one of them exceeds their corresponding limit, it would put the ratio over 1. So that would be a non-conformance. But in this case, all of the levels are less than their associated MCL, and it's a passing result. So when it comes to what NSF does as a testing agency, we evaluate products that come into contact with drinking water to ensure that they don't impart any contaminants, in this case PFAS, into drinking water from the materials that they're made of. For PFAS, the materials of concern are fluoropolymers, things that may have PFAS in their formulations. And the Joint Committee for Standard 61, which addresses products used in the municipal setting and water treatment and distribution, they are reviewing this and working on implementing that into the standard so that products can begin to be tested against that. I'm going to shift gears here a little bit and just talk about the EPA's rule and the timeline associated with that, and then talk a little bit about point of use and point of entry water treatment devices that can reduce PFAS exposure. So, as Doug mentioned, in April 2024, the EPA finalized their rule and set out an implementation period for when it will be enforced. Essentially, utilities and public water supplies have until 2027 to complete their initial monitoring and begin reporting levels of PFAS detected in drinking water. And then in 2029, enforcement begins. Public water supplies will have to provide public notice for PFAS found in drinking water, and remediation efforts will be enforced. So one of the first things that you may notice is that there's a gap of about 5 years between now and when this will be fully implemented at the public water supply level. And there's a lot of public concern around these contaminants right now. So the question we get most often is, what can we do in the interim? How can we help protect ourselves against exposure from these chemicals? So there have been, since about 2016, some test methods in place to validate point of use and point of entry water treatment products for reducing PFAS. So these are products that you can buy through retail, through dealership networks, online, or in stores, and use in your home. Right at the point of use, which means right before it's dispensed into a drinking vessel, or at the point of entry where the water main meets the house. So in 2016, NSF developed P473 to validate a PFOA and PFOS reduction test method. And this was somewhat in response to the health advisory limit that was set by the EPA at that time of 70 parts per trillion. So when you are talking about validating that devices can reduce PFAS, you have to also know at what concentration the PFAS is coming into the device to make sure that the public in general, 95, 99% of the public are experiencing contamination lower than what these devices have been validated to treat against. So at the time, we set the challenge level, the amount of PFAS that is going into the device, based on occurrence data from UCMR3, as well as a couple private wells. So I believe they were in Decatur, Alabama, and I'm blanking on the other one, but it was somewhere in New York. That were sort of hotspots where there were elevated levels of PFAS in well water that was intended for drinking. So shortly after that, the following year, this protocol that NSF developed kind of internally was presented to the Joint Committee and became a standard. It was incorporated into NSF standards 53 and 58. Standard 53 addresses filtration products. So this would be technologies that use granular activated carbon or anion exchange resins. And standard 58 addresses reverse osmosis systems. So that's the distinction there. And it was great for a while, but then public awareness and our understanding of PFAS began to develop more. And the Joint Committee got together and wanted to add more protection to the standards. So based on, at the time, various states were releasing their own MCLs. The Joint Committee decided to reduce the limits of acceptable PFAS in water from 70 parts per trillion down to 20 parts per trillion. And at the same time, as Bradley mentioned, the chain lengths and the different PFAS variations were also starting to become more widely known. And we wanted to develop something that ensured these devices were effective against a wider swath of PFAS. So several other PFAS were added, and we added a reduction claim called total PFAS. And then shortly after that was finalized and published, the EPA announced their rule and went even lower on PFOA and PFOS down to 4 parts per trillion. And they also regulated somewhat different variations than what ended up in the standard. So as it stands right now, the Joint Committee is reviewing the EPA's rule to incorporate some of these changes into the standards to make sure that point of use and point of entry devices are effective and aligned with the regulations. So on this slide, it's just kind of a summary of the differences between the current state of the standard and the EPA regulations. And we'll begin by talking about that influent challenge that I mentioned that was based on UCMR3 data and a couple private wells. Now we have UCMR5 data that's coming out. It's showing much lower occurrence throughout the US in drinking water. So right now we are challenging devices with essentially a sum of 2,160 parts per trillion of various PFAS based on the data at the time, but now we're seeing down at about 172 parts per trillion. So again, this is reflective of what 95% of the population would be in their drinking water. 95% of the population would experience lower than what we would challenge these devices with. This becomes interesting and important because when you challenge a device with this much PFAS, this 2,160 parts per trillion, it's much harder on the device to meet the criteria, and the overall capacity of the device is diminished by quite a bit. So it may not be appropriate any longer to be using these values. Some of the theories as to why it's so much lower now in the UCMR5 data is that, one, there's been increased attention on PFAS, and there have been treatment programs put into place. So overall, the PFAS is coming down. Another is that when it was under the UCMR3 data, the reporting or the detection limits were higher. So now we're seeing a mix of lower detections and higher detections. And finally, this UCMR5 data and kind of what's under consideration by the Joint Committee does not incorporate private wells where there are known hotspots. So those, or at least it's being discussed on whether or not those should fall outside of the scope of our standards because greater remediation efforts may be required in those cases. And our goal with setting these values in the standard is to protect the majority of the public without putting an undue burden on manufacturers that have to solve the worst case of the worst case contamination events. So this table in the bottom is comparing the UCMR3 data and the UCMR5 data, just kind of showing it in more detail. And also comparing the MCLs, or the limits that are allowed in the treated water. So under NSF 53 and 58 right now, actually Gen X is not even incorporated in there, so that's a small typo. But for the remainder, you see PFOA and PFOS at 20 parts per trillion, whereas the EPA MCL is at 4. And you see that there's a little bit of a mismatch between some of these. So, for example, PFBS is included in NSF 53 and 58, but it's not in the EPA rule. So there's a little bit of work to do to align with the EPA, both in terms of how much we're challenging the devices with, what the acceptable limits are for each one of them, and then what specific mix should be incorporated into the standard. So again, just in a quick summary, regulations change. PFAS has changed pretty quickly comparatively to some of the others. But the NSF ANSI standards are designed to change and be updated continuously to keep pace with regulations. Right now, there are task groups working on ensuring that materials don't leach into drinking water, that PFAS doesn't leach from materials into drinking water. And then also validating that commercially available devices are capable of treating PFAS and limiting personal exposure. But there's a little bit more work to do, and hopefully we'll finish that work up soon so that we can start promoting this to the public so that people have an option to limit their personal exposure to PFAS. That is the end of my presentation. Just want to say thank you, Doug and Mihao, for inviting us. It's been a pleasure.
Thank you very much, Kyle and Bradley, for speaking with us today. We do have time for questions. If you have any questions, please put them in the webinar chat. That way everyone can see them and we'll be able to get that out to, and we can get it noted. And for your information, this webinar is being recorded. So it will be available on the NEHA website in a few weeks, but if you have I just want to make sure you're all aware that it's going to be out. But if you do have any questions, here's a good time to do it. And the final, and from my end, I did get a few questions in originally that came in. EPA TSCA, this may be a question for Brad, but EPA under the TSCA, they came up with a standard. Would they get an even more stringent standard after these have come out? These rules get promulgated. Would they have the ability to even have a more stringent standard in certain circumstances? Brad, you're on mute. Yeah.
Sorry about that. I can only really speak to the MCLs and the MCLGs, which are the maximum containment level and the maximum containment level goals. I don't know that, I mean, certainly what we're seeing is that the EPA's numbers are going down over time. I mean, you can look at the EPA's health advisories from 2016. I think there were 70 parts per trillion. And now we see the health advisory down to I think it's 0.0004, if I'm not mistaken. That's the health advisory. Again, that's different than the MCL, but the MCL is 4 parts per trillion, which is still much lower than the previous EPA health advisory. And, because the current health advisory is so low that, it's not possible for, I'm not an expert on the detection, but I believe it's just not possible to detect as low as 4 parts per quadrillion. So the 4 part per trillion is based on practical considerations and what EPA thinks is a reasonable detection limit. But certainly we see these with EPA, we see these numbers going down. I think that's pretty consistent with other agencies. So I don't know. I can't predict the future, but certainly I think what we're seeing is these values continually going down. And I think there's a lot more of a, I think Health Canada has done this too. There's more of a, we're seeing more values get adopted that are based on detection limits rather than health effects, because these values based on the health effects are so low that we can't detect down to those levels. So we're seeing more of these regulatory values being based on practical detection considerations. So I don't know. I think I can't predict the future, like I said, but I think you're going to see more of these regulatory values be based on detection, our ability to the lowest we can reasonably detect, rather than a health-based value per se.
So they're basically unsafe at any level from the EPA point of view, but the MCSL would be the enforceable standard, and that applies to public drinking water systems, correct?
Correct. The MCL is the enforceable value. The health advisory is intended to be guidance, but is not enforceable.
With private drinking water sources, they would not be required to meet the MCLs, but does the EPA have any resources for them to use to test and perhaps clean up these their private drinking water standards?
Yep.
Brad, go ahead.
No, go ahead, Kyle. Go ahead.
I was just going to say, I don't know what the EPA does for private wells, but I do believe that there are testing
Kyle, did we
options.
freeze up there?
I think Doug maybe have you.
Yeah, you sound good to me,
Okay.
Kyle.
There we go.
Yeah.
If you could repeat that, I'm sorry, we froze up.
Gotcha. Yeah, the only thing I was adding is that I'm not sure what the EPA does for private wells or what sort of enforcement they do there, but there are testing options. You can send in samples and have them tested for PFAS, and then develop treatment options for those cases. Yeah.
Got a question online here. Will PFAS be incorporated into other NSF standards like NSF 61?
Yeah, so right now, there is a ballot being considered to incorporate PFAS, the regulated PFAS, maybe one other, into NSF 61 for materials of concern like fluoral elastomers.
And we got another question in here. It was asking about if people use but don't drink PFAS. If you have PFAS contaminated water, is it still a concern? Can you ingest it airborne, or is it a dermal problem, or is it only a problem if you drink it?
Well, I'm not an expert on PFAS toxicity as it relates to other routes of exposure. I suspect that with dermal exposure, my sense is that because there tend to be higher molecular weight, it would probably be a lower hazard compared to oral. I think oral is the main exposure route of concern. When you get these chemicals that are higher molecular weight, they tend to be less absorbable through the dermal route of exposure. And I don't think that these, based on the phys chem properties, there's low volatility, so probably less of a concern with inhalation. But that's not to say it's not a concern. If you get dermally exposed, there could be some limited absorption that would add to your total body burden, but I don't have any data in front of me to know what proportion of your total exposure that would be. So
Okay, so it's a concern really no matter what.
it would be a concern, I think, no matter the route of exposure, but I would suspect that your total body burden is probably going to be greatly, I guess, most substantially from oral versus dermal and inhalation, just looking at the phys chem properties. But
the difficulty in removing this for a small drinking water system, how difficult is it going to be, or is it something that's relatively manageable to do?
So in talking with the industry, it sounds like it's going to be a heavy lift across the board. There's a lot of concern about being able to do it, especially for underfunded, smaller community supplies. In those cases, though, having a point of use or a point of entry treatment product available is beneficial. And we're seeing increasingly that legislation will write in that when a public water supply is unable to achieve Safe Drinking Water Act compliance, that certified devices may be used to help achieve that. That's been the case with like lead in small and rural community supplies. So that's one possible solution, but in talking with the industry, my general sense is that it's going to be a heavy lift across the board.
Well, there's no easy answer to this anyway around. They're going to have to be doing some inventive processes to meet this standard. Did EPA come up with, I mean, they incorporate the difficulty when they come up with their MCLs. Is there an answer to do this, or is it something that's going to take, is it technology there, or is it mostly the problem is going to be mostly financial?
Yeah, I'm not sure. I'm cautious to speak for the EPA and what their plans are, their feasibility studies, but we do know that granular activated carbon ion exchange resins and RO reverse osmosis membranes are all pretty effective at reducing PFAS. So there are technologies available, and I think the burden will be the implementation of those, especially at scale.
And not to be too big of a plug for NSF filters, but for a private water user, what I'm sure you're going to say they're completely effective, but is that a viable solution for them?
Yeah, that's the whole reason the Joint Committee, again, that's made up of different manufacturers, different certification agencies, public health officials. They're trying to form the most reasonable solution for those particular cases where either there is no public water supply treatment addressing PFAS, or there's increased concern for whatever reason, to design the standard to make sure that they are effective, that you can rely on them, and that they're going to work.
Well, we don't have any more questions from this end. Final comments from either Bradley or Kyle that you'd like to share?
Yeah, I'll just say I've been impressed by the industry's work towards this. It's a huge problem, and I've seen a lot of very intelligent people, very driven people working on it, trying to find solutions. And hopefully together we can all kind of mitigate this big health crisis.
Well, thank you very much. I greatly appreciate this. Just to remind viewers that the PowerPoint will be available, that this recording will be available on the NEHA website in the next few weeks. There also is a survey that is attached with this. And if you could fill this out, that would greatly assist us in improving our webinars in the future. So we'd greatly appreciate if you can fill those out. With that, on behalf of NEHA, Bradley, Kyle, thank you very much for being with us this afternoon, speaking about this important issue. We are assuming that we got a lot of work in front of us here, and we appreciate everything NSF is doing and anything that all the drinking water utilities are up against out there. So with that, thank you. Have a great afternoon.